All right, everybody. Thanks for coming out today for Protein Design and Modeling Club. Very excited. We've got our sponsor for the evening is AI Proteins. We've got a bunch of swag, some mugs and stickers, bottle openers and koozies. So feel free to come get some AI Proteins swag. Very excited. We've got Phil Lorenz from Basecamp Research here. Phil's the Chief Technology Officer at Basecamp, which is actually a UK-based company. So comes from all the way across the pond to present to us today. So very excited to hear about what you've been working on. Phil, please take it away.
Hi. Thanks so much for having me, for organizing this event. Really excited to be here. I'm going to talk about data mostly, much less about maybe models or algorithms today, a little bit. But really excited to be here today and present some of the work that we've been doing at Basecamp Research. To kind of kick things off, I thought I'll give you a brief overview of our team because it's kind of a weird mix of people that we are at Basecamp. So we have some of us work basically on being on calls with Costa Rican ambassadors or going to embassies in South America. Some of us are just coming back from the Antarctic. Some of us are looking at weights and biases screens. And so it's kind of a weird mix of people working on kind of the same research questions. But maybe hopefully by the end of this talk, it kind of makes sense. I personally, my background is in genetics and genomics. And the main reason I'm saying that now is to kind of ask the computer scientists amongst you for mercy with your questions later. Yeah, but this is a brief overview. Basically, in a one or two sentence summary, we go around the world and try and sequence as much as we possibly can to build a big database of genomes and proteins. And so the main things I wanted to talk about today is why I think that's a relevant thing to do nowadays and how we're actually doing this, from permits and access to this data all the way down to the sampling, the sequencing, how we're organizing that data, and we're organizing that in a knowledge graph, how we're using some of that data for controllable protein design, and how we're trying to leverage some of the data for more complex design questions, for example, in gene editing and gene writing technologies. So myself being a bit more of a biologist as a background, the first thing I think is always kind of worth reminding us of is that everything that we're building, whether it's a deep learning model that leverages, I don't know, hundreds of millions of DNA sequences or amino acid sequences, or whether it's a biotech product application, in the end, it all kind of is derived from our knowledge of life on Earth. And that's been going on for a while, as you know, 4 billion years. And that's been kind of a running experiment where a lot of optimization has already happened to some extent. And I think one thing that's really worth bearing in mind is that, yes, we have somewhat of a knowledge of what's been going on life on Earth. But I think one thing that's really worth bearing in mind is that actually we don't know that much about it. One of the recent trends I saw when it came to some of the large language models is that people started drawing bubbles next to each other, comparing the size of these models or comparing relative numbers of parameters. And so I try to do the same about what we know about life on Earth and what we don't know. I think it's quite a striking difference. And there's basically, when it comes to the number of taxonomies that we have studied on Earth, whose genomes we sequenced, it's actually a very minuscule fraction of that. And so if we think about what models we want to build in the life sciences, how representative that data is, that's kind of one of the starting points, I think, to ask ourselves, is the data that we have available for what we want to do with deep learning model applications in this space relevant enough? Is it representative enough? Or is there something that can be done about this? So the first kind of issue that we are seeing to some extent with the data landscape in the life science is that There is probably 1 trillion species out there. I do think that number is somewhat outdated and there's probably a lot more. Those are estimates. I can't give you an exact proof for that number, unfortunately. There have been extensive studies on this about 8 years ago, something like that. Some of the more recent studies are suggesting it's actually more in the hundreds of trillions. So if anything, that number tends to go up with these estimates. But as I said, I'm unfortunately not able to give you exact proof of that. Now, when we actually look at the sequence data that's available, whether that's on the genome, the gene, or the protein level, another issue that I think is worth discussing to some extent is the level of redundancy of that data, and that it's not that representative. So if you think about Magnify, obviously hundreds of millions of proteins, UniProt about 250 million proteins. When you cluster these databases down, they shrink significantly. If you go from 100% to 90% based on the clustering ID threshold, you lose half the database or more than that. NCBI is similar on the protein level. I think actually on the genome level, one interesting stat about NCBI is 2/3 of all genomes on NCBI are from just 12 species. And if we think about the fact that there's trillions of species out there, again, I think that is an issue when it comes to the training data that we have available for what we're trying to accomplish. Now, the last point, that's probably some of the less familiar features that I'm not entirely sure if many of you have come across. This topic that's an issue that's being discussed very aggressively at the moment at COP15 at the United Nations. And that refers to what is called biopiracy. Basically, if someone takes a sequence of NCBI that has been, I don't know, let's say someone in Costa Rica sequenced a plant, they're doing some research, they're putting this up on NCBI, and then, I don't know, a few years later, a biotech company uses that sequence to make a product, they make profit of that. That is actually illegal, because if there's no tracing back to, let's say, the stakeholders in Costa Rica, that is against something that's called the Nagoya Protocol. Now, every country regulates that in different ways. Now, we'll talk about that a little bit more, but that is actually, I think, one of the key issues where regulation is really, really ramping up at the moment. The entire African Union is just demanding a 1% royalty on all sequencing data derived from the African continent, for example. And so the lack of traceability of the sequences that we have in the databases that we often use in deep learning is becoming a huge issue in Europe, a lot more than in the US, to be honest. And there are quite a few biotech companies in Europe right now that have to delete entire strain collections, for example, because of this. So it's something that is worth discussing. And I think with us being a very data-centric company, that's something that we're addressing. So to dive into that a little bit more deeply, and why I think this is important for us, even though, you know, we're, or the community here is a lot more concerned with algorithms, perhaps, and modeling of proteins. The Convention on Biological Diversity has set out in, I think, 2010 to 2014, what is called the Nagoya Protocol. What that basically means is that whether it's through an academic or a corporate research endeavor, whether that's sequencing or gaining biological data, the first thing you do, obviously, for that is you have to ask for consent for getting that data, whether that's speaking to a nature park, a land reserve, landowners, but you have to put in place a way to share the benefits that you're deriving from that data. And so whether that's training an AI algorithm or whether that's just taking the natural sequence and doing something with that, that is something that you have to talk to the person that is related to the place of origin of where that data comes from. And so the first thing before we talk about how we sample, how we sequence, how we're building a knowledge graph, is basically we have an entire team that's four members of our company that are exclusively concerned with getting permits and building partnerships with nature parks across the globe. And so basically what that means is they are talking to landowners, governments, representative of nature parks, scientific institutes, especially in South America at the moment, to build commercial partnerships. So they do know that this is going into a commercial entity, but they're aware that what that does is they are going to receive a royalty back from any commercialization that appears, and they're getting capacity building and they're getting data back for biodiversity monitoring, for example, to make sure that every single data point that's collected is in accordance with Nagoya Protocol, and actually many instances goes above that, which again is something that we can't trace back for the vast majority of public sequences, because even on Magnify, which has some metadata, I think something like half the entries on Magnify have the geocoordinates 00, because people didn't fill it in. So that's an issue, I think, when it comes to a lot of the data that we have available. And so we've decided to tackle that, to make sure that every single sequence, whether it's a protein, a DNA or a genome sequence that enters our database, can be traced back to not just the geolocation, but the consent of the landowner, access benefit sharing agreements, and commercial royalty agreements that we're making back to the nature parks across the globe. And once that's in place, only once that's in place, we actually go around the world. We have field scientists that are professionally trained in exploring nature. 2 of them have lived in the Antarctic for 2 years. They're still very fun people, but they've kind of gone through a lot there, I think. And just to kind of point out some of the locations that we've been to, I mean, the boat that you can see, that's actually a Point Nemo. I don't know if some of you are familiar with that location, but usually the humans that get closest to Point Nemo are actually astronauts, because it's so far away from any human life on surrounding islands. There's some sampling that's gone on in Iceland and volcanic places. That's kind of what we're trying to accomplish, because you can imagine that most of the data that's available on NCBI, UniProt, and so on is obviously derived from the model organisms that we have, and the Magnify is heavily biased towards kind of human microbiome studies to a large extent. And so really going out into some of these extreme places is something where we feel like we can address some of the redundancy that we see in public data. Oh yeah, of course, yeah. Yeah.
The kinds of genomes and protein sequences that you get from all these exotic places, when you look at redundancy and compare that to if I walk outside and swab the sidewalk and I pick up the bacteria from there, how different? You know,
it's, yeah.
If you filter for 90% redundancy, like are you
I mean, I'm going to give you an example later on in gene editing where we're struggling by basically we have to cluster way below. We have to cluster at 50%, and that's often not capturing it. So that's one of the main things. We were also wondering at the start when we, do we just sequence, a little, and a bit of the Hyde Park in London, and then is that it? And we do, and it is worth sequencing a lot of kind of mesophilic environment. There's a lot of diversity there. But the outsized benefit of really sequencing diverse places is really trickling down on sequence diversity. I think a lot of the questions we had at the start is how much more sequence diversity is out there? Have we already seen a lot? And I mean, after doing this for 2 years, I don't think we've even scratched the surface. Not even close. And there will be some visualizations to show that hopefully in a minute. Yeah. Hi.
Following up, your last slide really struck me. Do you find novel organisms growing in like freshly cooled lava?
Yeah. Well, the problem sometimes with metagenomics is we try our very, very, very hardest to be able to annotate this as a bacteria, but then all traditional taxonomic annotation tools, they fail to annotate this any further. So there are some issues, obviously, with taxonomic annotations with something that's completely vastly unknown. And the other point is that with the amount of horizontal gene transfer that's going on in the wild, you can have the same Plasmodium strain from 2 different locations, but a 10 times difference in their genome size because they've just picked up so many gene clusters and so on. So there's a lot of just gene sharing and HGT happening in the world that makes sometimes the taxonomies really, really hard. So we have to annotate on the genome level, on the gene cluster level, and on the individual ORF level in many ways. And they usually don't agree with each other in many instances. Yeah.
As much as it is exciting to go in these exotic spaces, would you consider biodiversity even in the human habitat? Even microbes have to actually evolve for a variety of things that have not seen in nature before humans. So how about tapping into that diversity? I mean, like,
microbial diversity.
Yeah.
Yeah. Oh, absolutely. Yeah.
Like microbes eating, plastics.
Yeah.
Just an example.
No, absolutely. I mean, we go to a lot of unnatural environments. We go into chemical wastewater spills, these sorts of things. We have some plastic degrading projects going on, absolutely. And microbiome data is also something that we are collecting. We're not doing human microbiome data because that has clinical implications. The regulations of that is tricky sometimes, but there's plenty to learn from all the other animals and other microbiome data that we're collecting as well. Yeah.
When you sequence this, how are you doing? These are just totally new sequences that we haven't seen before. How are you doing any sort of gene? Like, how are you constructing the genome, or how? A lot of these proteins are going to be longer than a typical reading. So
absolutely. So we have a complete metagenomics assembly pipeline. That's going to be one of the future slides, where I'm hoping to kind of explain why we're kind of combining multiple sequencing technologies together to then have both long and also high quality assemblies. But I'll go into that absolutely in a second, because yeah, that's exactly one of the key points to address. Because one Illumina read, I don't know, 200 base pairs. I mean, it's not going to cover very much. So you're absolutely right. That's something that we had to address. Yeah, this is kind of a quick summary of where we're at so far. We have tried to prioritize countries that have a lot of biomes, basically. So some countries are really, really diverse in terms of their biomes, others not so much. We're using the WWF definition of biomes. There's different ones. Magnify, for example, has their own biome labels. We're using those for comparisons. But some countries, Costa Rica being one of them, has the largest amount of biodiversity per space, basically. So that's one of the countries that we've prioritized, South Africa being another one. And we've launched over 70 expeditions, 20 countries in total. We're hoping that we're hopefully about 2/3 of global biomes by the end of the year that we will have sampled. Yes, hi. So that's the WWF definition of biome. There's around 350 or something like that of those. So it's like tundra or sub-Saharan desert or European coastlines. So those are the sorts of biomes. So they're not very granular, but that's kind of a good starting point when you're on the policy biodiversity level. On the data level, we use the Magnify biomes, which are hierarchical, and because we want to benchmark ourselves to Magnify in some situations, they can go a lot more granular in many ways. Cool. And then this is how we do metagenomics on the sampling side. We have to make an agreement with all of our nature parks to make sure that the sample that we take is not invasive to the ecosystem there. It's usually the size of a pea. Sometimes it has to be smaller. For water samples, usually you have to have, in some cases, 10 liters, which is, I don't know, 3 gallons, or I'm not so sure, but something like that probably. And then we sequence. As I mentioned before, we're using 2 different sequencing methods. Illumina reads are great because they're high quality, but they're very short traditionally. Nanopore reads are great because they're really, really long, but have slightly lower quality. So we're using both at the same time. We bring them together, and then we do metagenomics. The first step in metagenomics is actually to try and do an assembly. And my personal background is in doing human genomics, and so assembling DNA for a single species can be really, really hard. Sometimes for plants, it's extra hard because they're very repetitive. But metagenomics really gives you a pretty tough challenge. One of my friends who works in this quite a lot says, imagine you're doing single species assembly. It's basically like solving one puzzle. But if you're doing metagenomics, imagine you're in a puzzle store and a bomb has exploded basically, and you have to put together all the puzzles from all the puzzles back together. So that's kind of the problem, because you have tens of thousands of species, hundreds of millions of reads, and they all have to go together to form the right metagenomic assemblies for each species. That's usually done in what's called a De Bruijn graph, which is unweighted. That's where we link together the reads to check what can form an assembly. And the version of that assembly that is kind of the baseline of what forms a contiguous piece of DNA is called a contig. That's kind of the baseline on which we do annotations. And then our pipeline does a lot of annotations on various different levels. So on the microecology, looking at things like alpha diversity and certain biological features, we annotate genomes, we annotate ORFs. So we delineate the ORFs based on different gene models for different taxonomies. Obviously, if you think about eukaryotes, they have introns and exons. We have to annotate those differently to prokaryotes. We also annotate operons, BGCs, horizontal gene transfer events, also non-coding elements like promoters, tRNAs and other things, trying to do this at a large scale. And then that goes into a knowledge graph together with metadata. So every single sample that we collect comes with about 160 different types of chemical and geological metadata. So we measure not just pH, temperature, but also salinity, 30 different types of chemical measurements. And then depending on the environment, certain additional relevant feature like exposure to light and other physical or chemical measurements. And then we built a knowledge graph that our co-founder had the brilliant idea of calling it base graph. And this is kind of the data model, summarizing it very briefly, where basically proteins are linked to the ORF they come from, and crucially their gene neighbors, because in a lot of biological situations, gene neighborhoods or genomic context can give you a lot of information about their function. There's various different levels of taxonomic annotations. And when it goes to the sample, we can trace back the permit structure, the access benefit sharing information, the chemical data, and so on. So that's kind of the knowledge graph as a whole. To give you some key stats about this, we've sequenced about 20 terabase pairs. And the graph now, as of Monday, I believe, has 3.5 billion relationships. We have about 3x growth rate over the last 9 months. But one thing that we can do, which is really exciting, is that because we have a pretty good idea which protein families, in most cases, are derived or enriched in which types of environments, those protein families that we care about a lot, we can actually sample very, very specifically. And so fluorinases, which I'm going to talk about later, there's 2 biomes which we know pretty well. You can find an enriched number of fluorinases compared to your average biome. It's much harder to do for housekeeping genes, so like a DNA polymerase, you're going to find everywhere because every organism needs to replicate their DNA. But for certain specific metabolic or other types of enzymes that require something with respect to interaction with their context, we can use some of the metadata to be very predictive with our sampling. Now, one of the features that I brought up earlier with respect to the redundancy of public data, this is something that we kind of benchmarked us against NCBI and UniProt. When you do this clustering at 90%, 80%, and so on, and you lose the database size, we've done this in parallel. And you can see that going to these extreme places and trying to sample vastly different places actually means the database loss is a bit more mild compared to some of the public data that we have. Yeah.
It's a stretch, but I'm curious, if you are to look at specific habitat, or I don't know exactly how your definition of specific sample is, could you quantify horizontal gene transfer across different organisms and say, for example, by what you see as horizontal gene transfer in a given sample, distinguish that specific sample from that biome to a different biome. Do you see what I mean?
Absolutely. So we're using a lot of bioinformatics to map horizontal gene transfer. One of the things, that's actually a stat I should mention here as well. When you do metagenomics and you assemble contigs, it's very hard to assemble an entire genome of 10 million base pairs in a sample that has maybe hundreds of millions of species. So you have to assemble these contigs. Now in Magnify or other metagenomic projects, sometimes these are a few thousand base pairs long, maybe 10, 20,000 base pairs long if it goes well. We've made sure by combining these two methods that the majority of our ORFs are from assemblies greater than 50,000 base pairs. So we can really capture a lot of genomic context around that. And that also means we can often recapitulate an entire horizontal gene transfer event because they're usually not a single ORF, they're usually groups of genes. You can actually see a very nice symmetrical change in the coverage of reads across that. There's going to be an example later coming on where I'll show you a genome browser view of exactly that phenomenon. And it's actually something that happens a lot within samples. So you will see a version of the genome that had horizontal genome transfer and one that didn't have that. So there's even within samples retention and an execution of that happening. So it's hard to compare this across samples. It's often something that happens within a sample. Yeah, that's some of the key stats on the knowledge graph. Obviously, one of the things that's, oh, hi.
Have you compared the drop-off for different, with different species, like compare like prokaryotes, eukaryotes, and see that there are different contributors to kind of biodiversity?
Yeah. So what we've found so far is that eukaryotes seem to be a little bit more redundant and conserved. That's just because, as far as we know, the prokaryotic diversity is just way greater. And the rate of evolution in prokaryotes often is a lot faster. And so there's also a lot of convergent evolution happening, meaning that sometimes you can almost completely circumvent sequence space, even though you still have the same protein family. And so I think that contributes a lot to much less redundancy in the kind of ground truth sequence space in prokaryotes, I would say. Great. So one of the things that we care about is not just obviously proteins on a global level, but what do these proteins do? There's various different ways in which we can annotate proteins. PFAM is one of them. One of the kind of types of categorizing proteins that we're interested in are enzymes. And so there's actually a way of annotating enzymes that's been derived from what's called the Brenda database, which has been using these EC numbers, enzyme commission numbers. Nice thing about Brenda is that it's hierarchical. So it has these 7 super enzyme families: oxidoreductases, transferases, hydrolases, so on. And then it has a subfamily, a sub-subfamily, and a sub-sub-subfamily accordingly in a hierarchical manner. And so we've been happy to see that we have, in most of these families, more sequences. And so we want to think about, okay, is there a way we can leverage that for design? And so one of our collaborators, Noelia, she's worked on Project GPT-2, which came out, I think, about a year ago. We've worked with her on figuring out, okay, how can we make this controllable? And so probably a lot of you have heard of ProGen, that came out 2 months ago, something like that. We're doing something very analogous, but on EC numbers. And we're currently testing some of these sequences. Basically, what this does is you're prompting an EC number, and it generates a protein sequence that corresponds to that protein class. It's already up on Hugging Face, so if you want to give this a go, feel free to. We've looked at the structures for hundreds of thousands of these that were generated. They're not all great, but a lot of them are good. So that's kind of been encouraging to see. And since this is trained on Brenda, this is all public and can be used by everybody. One of the things, oh, hi, sorry.
I'm really curious for in your experience using all these methods out there for classifying enzymes. Do you see in your data sets that there's some chunk
Yeah, so for annotation is a whole nother beast. So for annotation, what we see is that a lot of the traditional annotation, it fails with public metagenomic data. I think in Magnify, there's hundreds of millions of proteins that are kind of part of the microbial dark matter. And maybe you can annotate this as a nuclear protein or involved in carbohydrate metabolism, but not much further. I would say it's pretty similar when using the same tools. So we have to use a lot of additional tools. It's not part of the talk. I'm not talking about annotation that much. One thing that we're using a lot is our graph embeddings. Sometimes what really helps is looking at the context in which you can find a protein. So things like gene neighborhoods and some biome labels. And if we use a graph embedding and then have a downstream task for annotation, in some cases, not always, can be really helpful. So we have to come up with kind of creative ways that where the sequence isn't always the key or the only element to do that with. Yeah. Yeah, and I mean this is showing one of the features in ZymCtrl, showing that we kind of can generate sequences that are kind of below the 40% sequence identity threshold, so kind of expanding sequence space to some extent. And then one of the key learnings that we had when we were training the model is that the more sequences you have in a cluster, so basically if you cluster down sequences for an EC class, meaning that if a cluster is really big, has a lot of representatives, means high levels of redundancy. And the more levels of redundancy you have in an EC class, on average, this is not a great correlation. There are lots and lots of exemptions. On average, it means that the model will generate more similar sequences to what's already been seen. I think that kind of intuitively makes sense. And so one of the things that we're doing is we're now fine-tuning this model on some of our own sequences. And we're testing zero-shot and fine-tuned sequences to hopefully finally get this paper out in the next, well, I don't want to make any commitments, but in the coming weeks or something like that. Yeah, one specific enzyme class I want to talk about are fluorinases. The reason fluorinases are really interesting is actually that there's a lot of fluorination happening in the medicinal chemistry kind of industry. A lot of the small drug molecules that we're taking are fluorinated. I think the last 10 years, something like 20% of all small drug molecules that came onto the market were fluorinated at some point. If you think about tomography, there's fluorination needed for that. And fluorination is a really nasty chemical process. The catalysts that you have to use are really horrible. I think most European and American chemical and pharmaceutical companies, they sent their substrates for that step to China, where it gets fluorinated, and then the fluorinated compound gets sent back. So it's a really nasty process. So the question is, if we were able to use an enzyme for this, this would obviously make this sustainable, much faster, more efficient process. But there's only a few dozen fluorinases out there so far, and they're not very good. They're very slow. They're not very thermostable, and they're just really not ideal for large-scale manufacturing purposes. So this was kind of one of our first mission that we sent our field science team to do, to kind of send them out to find more fluorinases. We found a few more natural ones in kind of a big kind of discovery campaign. That really helped to have this added diversity. The vast majority of public fluorinases are within 85 to 90% similarity to each other. And that's obviously, if you think about, can we expand sequence space? Can we generate more? Using that as a training set is somewhat suboptimal. And so there's one example here where we have a fluorinase from our knowledge graph. That's a natural one that only has 30% sequence ID to any previously known fluorinase. But the structural conservation is actually quite good. So just to show that we can identify some of these. We then did some testing on this with LCMS to fluorinate a specific compound. And we tested about 200 fluorinases, some natural and some designed ones. 93% of them expressed, and about 2/3 performed better than the previous controls. and then some of them actually performed 8 times better. So we still have to push that a lot higher. So the ideal one would be trying to get to 30x. So that's still ongoing to improve this further. And then we also improve thermostability because that's really important for some of the kind of large scale manufacturing purposes. But that's kind of an enzyme example where without the added kind of sequence-based diversity, we wouldn't have been able to design some of these fluorinases that had this higher performance.
Was the improved thermostability a natural enzyme or something that you designed based on the new natural?
Both, sorry. Both, yeah. And then there's an EC class for fluorinases as well. One of the things that we were a bit surprised by, we fine-tuned ZymCtrl, which is trained on Brenda database, which is basically interlinked with UniProt. So we filtered out any overlap between NCBI and UniProt and fine-tuned it on the remaining NCBI sequences. And it actually looked like it produced worse sequences. And that's actually, we had a look at them and it looks like a lot of them were actually chlorinases. So it does seem like there's a bit of an issue. And we're testing some of them as a control now to perhaps recharacterize them as chlorinases. And then fine-tuning them on some of those has given us a lot higher PLDT scores. Again, kind of showing that the additional sequence diversity of the natural fluorinases that we found has really helped with that. Great. Kind of final example I wanted to talk about is that for a lot of kind of protein design challenges, the amino acid sequence or the structure of a protein, kind of the intrinsic nature of the protein is not kind of sufficient to solving what you're trying to address. And so, I mean, some of the kind of big hit examples for that, obviously, kind of CRISPR systems and nucleases where you need to have an associated guide RNA, for example. Another system that was made public recently are large serine integrases, which are basically a way of enabling gene writing therapies. And so I want to focus a bit on the latter, on which we've done a little bit of work to kind of showcase how combining protein with genomic information can be leveraged to solve some of the design challenges. And so the ARC Institute, at the end of last year, they published a study on large serine recombinases. I don't know if many of you are familiar with this type of enzyme. Basically, what it does is it's a type of enzyme that sits in a phage genome. So phages are types of viruses that kind of attack bacteria. What it can do is the phage will kind of go into the bacterial cell, and the integrase that's encoded by the phage will recognize two attachments, will recognize an attachment site on the bacterial genome, and then will integrate the phage genome into that site, and therefore splitting the bacterial genome up. And then within these two attachment site halves, you will then see a phage genome insertion. The reason this is a really, really attractive system for gene writing is that if we understand how this protein finds the attachment site and what that attachment looks like, we can basically write genes into the human genome. And that's what a lot of gene editing companies are currently working on. Because a lot of genetic diseases aren't just solved by addressing a single edit with a CRISPR nuclease. Some cases, you have chromosomal aberrations where you have, half a chromosome are missing. And therefore actually writing DNA into a genome rather than just making a small edit is a really promising type of technology. And large serine integrases are one way of potentially addressing this. Now, the paper did a kind of a bioinformatic mining of these from NCBI, and this is showing on a T-SNE representation large serine integrases from all of NCBI at 50% clustering. We did the same on our database, and this is now going back to some of the earlier questions of, is it worth sequencing more natural diversity aren't you finding the same things over and over again? And at 50% clustering, we're filling out a lot more extra sequence space. So every dot is a cluster representative, and it's about 30 times as many clusters at 50%, and obviously covering a lot more of this natural diversity. Hi.
So if they're so different from the NCBI representatives, how are you calling them as
So this is based on the. I mean, we did a like for like comparison. So it's the same procedure that NCBI did. That so it's based on a combination of PFAMs. Yeah. And then I mean the other point to make is that the other key difference is obviously that every single one of the LSRs that we have isn't just different in terms of the sequence space that they represent, but they actually are a form of a network where they're linked to taxonomic information, genomic context. The reason I'm bringing this up again is because this protein, we only understand how it works if it can attach to the right integration site, and this is where some of the bioinformatics is really important. Sorry, I forgot I had this slide. This is showing the same thing as a phylogenetic tree. But yeah, on the integration site, just knowing the amino acid sequence or the structure of the integrase is not really sufficient for a gene writing application. And therefore, we have to figure out, okay, when we see one of these large serine integrases, can we identify where it has integrated into, and can we therefore extract what the integration site looks like? And is there anything similar in the human genome that would enable this writing technology to be applied to therapeutics? And so this is looking at a genome browser, and this is actually going back to some of the horizontal genome gene transfer questions earlier. When we look at all the reads that are aligned in this metagenome, you can kind of see a symmetrical shift. There's a little kind of gap in the middle. That's the phage genome. And you see different coverage on either end. That's fairly symmetrical. This tiny blue little rectangle, that's the serine integrases. And so you can see about 1 kb upstream of that. That's where the bacterial genome starts. And then this way onwards, we have the other half of the bacterial genome. And if we separate these pieces of DNA out and annotate them taxonomically, we have a phage annotation for the insertion, and the rest is bacterial. So it makes sense on the bioinformatics side, but also on the taxonomic side. This is showing one example where we had a 7 kb double-stranded DNA phage genome inserted into a Clostridium genome with a serine integrase. And the structure of this LSR that we found is somewhat similar to kind of one of the studied references, BXB1, and the sequence ID is really, really low, again, showing that the vastness of sequence space can really be identified through that. We've done this for about 20,000 of these attachment sites, and we're currently mapping them to the human genome to then identify exactly whether those are what's called safe harbor sites, because you can't just write DNA anywhere into the human genome. That could be an oncogene or really important gene, so you have to make sure it's somewhere in the genome where that integration would be safe. And that's one of the kind of follow-up steps. But I think it shows a nice example of where whether you can design a protein or not, whether you can optimize a protein or not, looking at the context and leveraging some of the kind of genomic data that are surrounding the proteins that we're working on can be helpful to optimize this for some of the challenges that we're trying to tackle. So with that, there's kind of two summarizing thoughts that I wanted to share, which is that the first one, which I really care about, because that was one of the key acts of criticism, perhaps, that we were kind of faced with at the start, especially from investors, which were, are you positioning yourselves against generative AI, or do you guys hate generative AI, or any of that? And I absolutely don't, and I love working with some of our collaborators on AI. I just think the framing of discovery versus AI isn't really right. I think discovery can enable a lot of AI. We can use a lot of AI to guide our discovery. And so I actually think that they are beautifully complementing each other. And the other point is that it's been a real privilege to partner with some of the people that take care of our planet's biodiversity. And on that note, just want to say thanks, everyone. And if you have any more questions.
So I just want to see if maybe actually there's a microphone. We have a microphone now for questions.
Cool. Great.
I just want to see if I was following the last part about recombinases. Yeah. So you were looking at a bunch of sequences through, all of the collections that you've sequenced that had phage insertions in the genome, and so you could identify those, and then because it's the phage DNA that's inserted, you knew the recombinase because it's coding for that, so you can map where it's inserted and the recombinase that does the job because it's in the insertion.
It has to be that recombinase.
Right.
Exactly.
Okay,
yeah.
I see. So that's the main advantage of having diversity in the.
Exactly.
Okay, thanks. Any more questions for Phil? I have a microphone we can pass around.
I can speak loudly.
We pass that.
Are there plans to make your data available for academic or nonprofit use under some sort of license?
We can't, because we've made agreements with biodiversity partners, and a lot of them have said very, very loudly that they will never allow any of their data to be public, because there are tons of situations where, as I said earlier, someone has sequenced a piece of soil in Tanzania or a plant in Costa Rica that has gone up in NCBI. Someone has sequenced, has used those NCBI sequences, derived some small molecule information for that, made $1 billion drug, and no one has shared any of that with the people that have taken care of that biodiversity. And so our contracts aren't allowing us to do that. We can do that for specific protein classes. So we are working with some academics, including Alex Bateman, the head of UniProt. So we are working with some academics on specific protein classes or fine-tuning models with our data. Some of that can be made public. It's completely fine. But the database as a whole, we can't make public.
That's so interesting. Then if I could ask another follow-up question, because in the context of software and licenses, there's types of licenses where, if you make something open source and everything based on that has to also be open source. So I guess, do you see going forward, because this seems to me to be just such a critical part of biological discovery, to just try to make more frameworks where we could have better sharing of sequencing data?
Yes and no. I mean, that's going deep into policy. I think one way in which that's possible is that there will be certain types of applications where that can be done for. And so, for example, I don't know, something like plastic degradation, the hydrolases related to that function. We're now working on that a lot more, and that's going to happen in collaboration. I do think that, and I think that's perhaps not something that we're really aware of, the degree to which some of these countries have been screwed over by Western science, I think is maybe not fully appreciated. And so there is really a lot of very harsh resistance to the idea of open sourcing. A lot of the people in this room, myself included, I'm from an academic background. I think open sourcing is the most important thing ever in academia. If you speak to a nature park in Tanzania, they think it's one of the biggest detriment to the economy. So it's something that we have to take into account because we work with them, and in many cases we are the only people they trust to do this work with. And so there are some considerations to kind of balance this, if that makes sense.
Thank you.
I have a couple of questions. So I would imagine a lot of these unique, what should I say, like microbiota you will find in the oceans. Do you plan to do something which is like going deeper and deeper in the oceans?
Absolutely. I've actually spoken with some of the audience about exactly that. We've done some kind of shallow diving, but we have some plans to do this more extensively and also actively looking for collaborations. We have our first deep diving expedition at the end of this month, I believe, or early next month. So absolutely.
And the second question related to that is sort of who owns the ocean if it's beyond, let's say, 12 miles from the coast?
Yeah. So I mentioned the protocol. That's mostly soil and land. There's something called the BBNJ, which is a treaty on the open seas that was not ratified, but has started to be negotiated 2 months ago or something like that. No country really owns all the seas, and there's lots of regulation around that. There is currently a suggestion, if I understand correctly, for a mechanism where this will be organized by the United Nations, and then some of the access benefit sharing goes through the United Nations like a clearinghouse and then back to the individual countries. But there's lots of negotiations still happening, and how that is going to work mechanistically is still very complex. What's good is because we have an entire team that's only doing this, so about a fifth of our company is only doing biodiversity work, contractual obligations and all of that. 2 of our members are actually part of the Convention on Biological Diversity at the United Nations, so they're constantly there to be updated and be part of that regulatory framework. So that's not settled, basically. That's the answer in short, I would say.
Thank you.
Hi, Philip. Great talk.
Thanks.
When I think about the idea of sequencing things across all these different biomes, I often think about that for the overall goal of like thermostability or increasing stability. So you told one story where you were able to incorporate a few mutations to increase stability, but I was just wondering, are you able to do that with all of your protein? Like, you're sequencing and then expressing these proteins? Like, what % are you able to like experimentally characterize? And then within the scope of stability, what are those parameters that you're looking at?
Yes, so we're not characterizing every single one of our protein. That would be a lot. What we're doing is we're doing this kind of in a very bespoke manner internally. Like we did with fluorinases, we're doing this on some of the nucleases, we're going to do this on the serine integrases, where we have a kind of a case study that we're trying to display some of the data advantages, or where we're figuring out, okay, to what extent does metadata help us to make a prediction, or can graph embeddings annotate this dark matter protein? So we're working on that internally, and we're doing expression and testing and that with a COO. Everything else is tested by our customers, basically.
I just had a follow-up to the previous 2 questions. Actually, to the ocean question, how much similarity is there between an ocean sample and the Tanzania soil sample?
That's hard to quantify. I mean, one thing you could imagine is if you go outside this park, outside Harvard Medical School, and you were sequencing it tonight, and let's say it's going to be slightly acidic rain today, and you sequence again tomorrow morning, there's a good chance that 80% is different. So the amount of change and death of species that happens within a few hours in a metagenomic sample can be really rapid. But you can also have really stable environments. So it's very, very hard to quantify this on a geographic scale, let alone on a time scale as well. So I can't give you a quantifiable answer to that. We've had situations where there was surprising overlap between 2 somewhat different environments in Finland and in Spain, which you would argue would be, have slightly different climates and so on. But it's very hard to always be 100% predictive with that. For certain protein families, it works really, really well. With iron-rich soil, you're going to see iron-sulfur clusters a lot more. That makes sense to some extent. There's also some protein families where that's a little bit more obscure, but these relationships work. And I don't want to mix causation and correlation, but it helps with our sampling. And then there's some relationships which just don't make sense, or we're not yet really able to mechanistically explain. what these differences are about.
Maybe one follow-up before
Yeah.
I give the microphone back. I think this is really cool. I've seen like 10 or 15 years ago, there was a talk by Craig Venter, who's done something like this, Sorcerer 2 expedition. He wanted to take a boat trip, and every 200 miles in the ocean, he took a sample and said they're completely different. And he was complaining, I can't deposit it in NCBI because I would double their number of sequences. And do you know anything what became of that?
We've spoken with Craig a few times, yeah.
Okay, thanks.
But I mean, we don't have them. No, we don't have them, no. Because again, we need to have everything from permit and ABS onwards. For these samples, I know it's different. But yeah, we don't have the samples. No.
So maybe a bit of a challenge and a curiosity. In a sense, when you get so much data, you probably don't know what you don't know in there. In a sense, could you have in this data set, these sequences, enzymes that catalyze completely novel reactions that we had not known? How would you go about digging that and identifying that? Becomes another challenge, I guess.
Yeah, that's exactly what I would say is probably the most interesting question to ask to our knowledge graph. We're trying to attack this problem from multiple different angles. Some of this you can kind of try and think about, are there similarities between, I don't know, if it's about a chemical conversion and you have an enzyme reaction that involves element X, is there a reaction that is perhaps similar enough to catalyze something that involves something that is, one field below in the periodic elements? So like there's certain things where if you consider atomic radiuses and these sorts of things, where you can start making some assumptions on very specific cases and then go from there. That's one kind of more kind of biocatalysis type way of addressing this. Contextualization can sometimes help. And then we leverage the metadata. So both on a kind of generalizable way with graph embeddings, but then also sometimes if there's, really, really, really high levels of molybdenum in a piece of soil. Molybdenum being a frequent, cofactor in a lot of kind of biological pathways. That's also sometimes where you can start drawing some conclusions, but it's very hard to address this generalizable. There's obviously things like protein fur, which have annotated more sequences with the PFAMs that we already have, mostly. That's also something that we're leveraging. But to really truly identify somebody wacky that's never been seen before, we're trying with a couple of different avenues. But I don't have a satisfying, all-encompassing answer for that, unfortunately.
So we've got one question from the folks online. I think you've kind of answered this before, but I'll ask it anyways. Is any part of the knowledge graph and new sequence annotations available online somewhere, or are these confidential?
There are going to be some for ZymCtrl. So we are looking at 2 enzyme classes specifically, where we are testing them at the moment in the lab, of natural sequences kind of as a control, zero-shot generated and generated after fine-tuning. And so the natural sequences that come from our knowledge graph, they will be made public for those 2 protein classes. But as I explained earlier, we can't make larger parts of the knowledge graph available. We are starting some academic collaborations, as I mentioned before. There's like one group that works on, porins and membrane proteins. And as part of that, those sequences, some of them will be made public. So on specific case-by-case basis, we are starting to work with academics on specific protein classes and related sequences. They can be made public as part of that, but as a kind of large-scale generalizable thing that's going to happen, we can't.
Phil, I have a question about if all of the non-redundant sequences could be used just straight away in a model like AlphaFold to, make a better MSA for a certain protein, and maybe it becomes a better, more confidently predicted structure. Have you seen just drop-ins, like, oh, we can make better MSAs for proteins because our sequences are more diverse, or?
Yeah, absolutely.
Is that right?
Yeah, absolutely. In some cases, the problem is that we have to do some curation of the MSAs because sometimes it gets so far away that it can't build an MSA, or it's too big or something. So we have to do some pruning sometimes. But yeah, absolutely. That's exactly what we're trying to leverage with that.
Could you imagine giving people not access to the MSAs, but behind some, firewalled thing that AlphaFold can fold their sequence, but they never see your proprietary sequences?
I could imagine a solution for some of this happening, and we are working on it. We're not working on a folding question at the moment, but a more kind of language-based annotation method at the moment where that could be a consideration. But this is also, I'm not the decision maker on that. I don't know what's going to happen with that, but okay. It's an option. Yeah.
Thanks.
Okay. There's one more online one, sort of along these lines, is how are your partnering and collaboration policies?
It depends on the protein family a lot. So it's usually very helpful if that can be linked to a specific class of protein or taxonomy, or I don't know, a gene cluster or something. But yeah, it depends. What has to be made public. In general, we're super open. We've started with a total of 5 academic groups right now that some of the work has already started, some of them in discussions. So we're really, really open to that in general. There's lots to be discovered, I guess, and studied. It's just that when it comes to making large parts of sequencing data or annotated data public, that's the issue, but there are ways to address that. And again, for specific protein families, even that can be possible.
I got the mic once more.
Yeah.
You mentioned PFAM and the ongoing annotation work. How often is PFAM out of its wits with how often is PFAM, how often is it unsuccessful in figuring out the protein family? Maybe I missed it, maybe you said it.
Oh, as in what proportion of ORFs have no PFAM, basically?
Yeah.
I'd say we're pretty similar to magnifying that. So I think it's about 1/3 to 40%. If you go to kind of the microbial dark matter, it's quite a large portion that's hard to annotate with a PFAM. But that might be more like 28. I don't know the exact number, but around that kind of scale, I think. Cool.
All right. Any last questions? Maybe I'll take the privilege of being the organizer and ask one more. It just seems like there's a lot of environments where there's the potential for, I would say, convergent evolution, right? They're perhaps have similar conditions, but are on really different parts of the planet. I don't know how much you've gotten to look into your sequences. Like how common are you able to see examples of convergent evolution?
Absolutely. That's it's actually been posed to us, that question, by a customer of ours, actually, in the cosmetics industry. I didn't. That was a bit of a curveball, but basically they were looking on a catalytic conversion on a specific substrate on human skin, and if you look at the human skin microbiome, vastly different species. Some of them will have evolved to catalyze that substrate, and so you can have 2 strains that have for that enzyme class, protein class, 99% sequence similarity. One of them can catalyze it, the other one can't. Or you can also have 17% sequence similarity between 2, and they both can catalyze it. So like the kind of sequence to function relationship on that kind of more complex kind of convergent evolution is quite tricky. But graphs have kind of helped us with that, contextualizing taxonomies in that regard. When it comes to kind of a more global environmental scale, we haven't leveraged that as much, probably. But I think on the second we can kind of zoom down on individual protein families, that's where we can sometimes start making some conclusions in that regard. And then we kind of look at structures, and then if there's if the active site looks similar enough, then you can start making some further assumptions that can be helpful.
Awesome. Awesome. Thanks again, Phil, for coming in. A great presentation. And thanks everybody for an awesome discussion. We've got more pizza and beer, so feel free to hang out and talk to Phil some more.